Hello everyone, my name is Fedor and today I'm going to share with you something very special about our latest AI model, P3GPT. P3GPT is not just an AI system, it's a live model which can digitally emulate the results of any omics-level experiment you can think of. That is, now you can easily, cheaply, and quickly obtain the results for your omics hypothesis if you just run P3GPT on your computer. And today I will share with you how exactly you can use it to power your aging related research. So first, this notebook you can find on our Hugging Face repository. You first need to initiate the handler object, which allows you to communicate with P3GPT. And what we will be doing today is we will be studying cross-species aging hallmarks, that is, genes whose regulation is important in the context of aging for both mice and humans. And we will do run generations for 3 different tissues, and we will consider both genes that become upregulated from the in terms of RNA expression, and which are also regulated in terms of epigenetics. That is, with aging, these genes promoters become hyper-methylated. So first you start with designing your prompt, which is structured and really easy to edit. Just make sure that you fill in all the fields that are relevant for your experiment. You obviously will need to have a table to store the results for all your generations. We will be running 12 different generations today, comparing young mice and old mice, and young people versus old people. And now I will quickly start the generations. It will take a couple of minutes, hopefully not so long, but with the magical power of editing, I will be with you real shortly. Now, our generations have been completed in a little bit under 2 minutes, and now you are free to explore the generated aging signatures across multiple species, multiple tissues, in whichever way you like. But one thing that can lead to some interesting discoveries is inspecting the genes that are shared in the upregulated domain, in the upregulated hallmarks in both humans and mice within one tissue. Now we can actually see that for every generation. We asked P3GPT to provide a list of 100 most important aging genes in each tissue and each species. And it turns out that 33 genes are shared in the aging signatures of livers from both mice and men and humans. Now we can take a closer look at these 33 genes. We can take them to whichever analytical pipeline you like most, but my favorite one is Enrichr by Mayan Lab. And Enrichr is a wonderful tool which allows you to very quickly, visually, and informatively get annotations for your gene lists. So I will select some human, some murine databases. I will insert my list of genes here. All 33 have been recognized, and now we can see whether any of these genes form hubs. So some of these genes are connected to just 1 or 2 terms, nothing interesting, but here we have a very dense cluster of genes with some complement proteins, NAT metabolism proteins, and other proteins which are connected to many different categories. What categories are those? Well, we can see that some of these categories are actually relevant in the context of liver. We see some liver diseases, we see human liver hepatocyte specific genes. We also see some urine liver proteins, but also some unexpected cerebrovascular disease genes as well are present in this list. So what could that mean? Where do you take this further? Where at In Silico Medicine, we love employing agents, AI agents, to run annotations for such tasks which involve a lot of routine search operations and digging through lots and lots of literature. Actually, we have numerous agents for every task one might need. One of them has been created by my good colleague Anastasia Schneiderman, and the tool that she has created allows us to upload a gene list and ask AI to describe these genes in the context of a disease. For now, let's specify aging-related liver diseases, and all that's left is to ask the model what is the role of these genes in liver aging? Question mark. We'll need to wait a second while the model searches the web, finds the sources to give us some relevant information about the gene list that we have just uploaded. So we selected some of those hub genes that MindLab identified, and yeah, we see that some of these genes are described in the context of non-alcoholic fatty liver disease and other aging-related conditions. Now we can select any one of them and take this analysis one step further. You can open DORA. DORA is our new tool to write scientific academic texts, and not only that, and back your academic text with relevant literatures, actual references from PubMed, and we have a number of templates that support various types of scientific texts. But for now, I'll specify this as aging-related diseases. Actually, I'm not sure you see it properly. Let's zoom in and let's provide some context to the model. So Enrichr-KG has identified AHSG as an aging hallmark gene in both murine and human liver. And one of the most recent features that we have added to DORA is that you can upload your bibliography directly into the text generator, and this way the AI system will pay attention to the information sources that you like. So previously I uploaded the Precious 3 preprint paper that we have deposited on bioRxiv, and here it is. We can attach this paper as a reference for all the text that DORA will try to generate. So first you have an outline here of the paper that will be generated. We do not want to change anything, although you could provide some extra context for the model. Now we'll need to wait a short while before DORA generates a more detailed plan of the review paper. You can inspect that and click generate to get the full text of this paper, but to save us some time, I have already run this prompt in DORA and got this wonderful paper. So you can inspect all the literature that was referenced by DORA in producing this text. You can study how our target gene has been annotated in various contexts of fatty liver disease, fibrosis, liver cancer. You have all the different sections here, which involve other tools which are integrated with our other products such as Pandomics, Precious 3, and in the end, you can modify this text, present it to your colleagues, or maybe use it for your own research articles. And basically, that's all I wanted to show you. Today, we started with an interesting hypothesis, an interesting research question, which genes are important for aging in both humans and mice. Then we asked P3GPT to generate these genes by emulating a case control study between young individuals and old individuals. We took these genes to Enrichr-KG to run pathway analysis and enrichment analysis in different research databases. We took this information, identified some interesting potential targets for geroprotection, employed AI agents to see which genes are relevant in the context of aging-related liver disease, took one of those and employed DORA to write us a complete review article about the role of this target gene in aging and liver pathology. Now, the whole process takes less than half an hour. And this is the power of AI, in which we deeply believe here at In Silico Medicine that AI allows us to move at a breakneck pace with our research of clinical targets, molecule design, identifying novel hypotheses, and now with P3GPT we can run large-scale experiments at a fraction of the cost, a fraction of the effort, and a fraction of the time period in which it would take to run these experiments in proper animal laboratories. Now, I hope you've learned something useful for your own research and you will try P3GPT to power your own research endeavors. Okay? So that's all for today, and I hope to see you sometime soon with even more exciting demos of P3GPT. That was Fedor from In Silico Medicine. See you. Bye-bye.